Hello and welcome to all my dear lovely students. I hope you all are doing fine and must be studying well at home. So once again I welcome you on this amazing platform of Physics Wallah in your excellence batch, where we have started the second chapter of class 12, that is sexual reproduction in flowering plants. Already we have discussed first two lectures that were mainly related to that how the male sex organ, that is androecium, results in the formation of male gamete, and what is female reproductive organ, that is carpel. Today we are going to study that how in this carpel the actually female gamete is prepared. So today's lecture is all about to understand the types of ovules, that is Megasporangium, and how within Megasporangium Megasporogenesis takes place. So without a delay, let's start with today's lecture. In my last lecture, I have already told you what is ovule and the structure of a typical most common ovule, that is anatropous ovule. Apart from this, there are many more types of ovule depending upon the position of funicle and chalaza and micropyle. So in total there are 6 types of ovule, which here one by one I will be discussing. Please stay connected. So first, on the basis of position of micropyle, chalaza and funicle, there are 6 types of ovule. First, let's start with the most common ovule observed in 80% of the angiospermic families, called as anatropous ovule. So it is the most common. 80% of angiospermic families, they have this type of ovule. Correct? It is actually the inverted ovule. Why? Because the distance between micropyle and funicle is least, and chalaza, which is actually the base of the ovule, seems to come at the top. I'll show it also with the help of the diagram. But before that, let me write the theory and then I'll draw the diagram. So for anatropous, what will you write, beta? Chalaza is at the top, whereas micropyle and funicle are at least distance. They are minimum distance. They are very closely present. Are at least distance. So how will you draw? There are 2 ways to draw anatropous ovule. The one which I have drawn in my last lecture, like this. Right? So these are the integuments. This is the chalaza. This is the opening, that is micropyle, and this is the stalk, which is funicle. So you see chalaza is at the top and the distance between micropyle and funicle is minimum. They are very close to each other. One is this way to draw the anatropous ovule, and second way is this. Again you see, beta, this is the micropyle, this is the funicle, and this is the base, chalaza. Correct? Now you will say that ma'am, chalaza is at the base. How this can be anatropous ovule? So beta, main thing you have to see the distance between micropyle and funicle. If you see that the distance between micropyle and funicle is minimum, that means it is anatropous. It is inverted ovule. So both the way you can draw in your NCERT. If you look this diagram, this is A, which I have drawn in my class, and this is B type of anatropous ovule, which is given in your NCERT book. Correct? So please make sure that you understand this type as well and don't write that it is a straight ovule. Right? Now, second type of ovule is orthotropous, which is also known as straight ovule. It is the most primitive one, which is observed in, for example, cycas, gymnosperms. Gymnosperms are the first group of plant kingdom beta to form ovule, and their ovule is the most primitive one, which is straight orthotropous. Here, the distance between micropyle and funicle will be maximum, and chalaza will appear at the base, micropyle will appear at the top. So, what is orthotropous ovule? Distance. Main point is to check the distance between micropyle and funicle. So, in case of orthotropous, distance between micropyle and funicle is maximum. Both M, F and C. M stands for micropyle, F stands for funicle and C stands for chalaza. All the three lie on the same straight line. So you can say all the three, they are coming at 180 degree. I'll show you this with the help of the diagram also. Have you seen the logo of LIC? How it is? It is like this, na? The logo of LIC. So orthotropous is also like the logo of LIC only. Okay? These are the integuments. This is the opening, which is micropyle. This is the base chalaza. And here is the stalk of the ovule called funicle. So you can see micropyle is at the top, funicle is at the base, and all three are coming on the same straight line. So this is your orthotropous ovule. Is it clear? Both anatropous and orthotropous. Okay. Moving towards the third type, which is hemianatropous. The example of hemianatropous is Ranunculaceae. Ranunculaceae are the primitive angiospermic families. So in primitive angiospermic families like Ranunculaceae, the type of ovule is hemianatropous. Ranunculus. Now what happens, beta? You have seen an orthotropous ovule is straight, but in hemianatropous, ovule is going to bend in 90 degree. So funicle over here, chalaza on this side, micropyle this side. So the distance between micropyle and funicle is going to come at 90 degree. Such a ovule is called as hemianatropous. So what is hemianatropous? When micropyle comes at 90 degree to funicle. Let's see with the help of the diagram how will it look like. So now this is how the diagram is going to appear be. Beta, this is the opening, which is micropyle. Here is the base chalaza and this is the stalk called funicle. So actually it is at right angle. Correct? Micropyle and funicle, they lie at 90 degree, and here will be your embryo sac. Correct? The third type of ovule is hemianatropous, which is seen in primitive angiospermic families like Ranunculaceae. Moving towards the next type of ovule is chempylotropous. So, next type of ovule is chempylo. Chempylo means angle even less than 90. Again, see, this was orthotropous. This is hemianatropous. Now, you bend it more. Bring micropyle more close to funicle, decreasing the angle from 90 to acute. This type of ovule is known as chempylotropous, which is seen in families like Leguminaceae and Brassicaceae. These two families you also study in detail when in your chapter morphology. So examples of chempylotropous are members of Leguminaceae, that is your pea family, and Brassicaceae, that is your mustard family. Okay, brassicaceae spelling. Right? Now, what happens in Campylotropus beta? The angle of the ovule is going to get reduced. Can you see how I have turned my micropyle? So this is my micropyle, this is chalaza, and this is funicle. See, angle has come less than 90 degree. Understood? Initially like this, now again it is bending. So this type of ovule is known as Campylotropus when micropyle and funicle are at acute angle. Okay. Now, next type of ovule is Amphitropus. Amphi beta means both. Here, the shape of the ovule will become horseshoe. Horseshoe, you know, like this. This is horseshoe magnet also you study. So, here in case of Amphitropus ovule, both the ovule, the shape of the ovule and the shape of the embryo sac is going to become horseshoe. As a result, it is known as Amphitropus ovule. For example, in lemna, in poppy, in case of elisma. So, examples. First, let me write both ovule and embryo sac becomes horseshoe shape. I'll show you with the help of a diagram, but first write the examples like elisma, poppy and lemna. So how are you going to draw it, beta? Just see it. Right? This is how you will draw. This is the micropyle, this is the funicle and this is the chalaza, and again the embryo inside, the embryo sac has also become horseshoe shape. Right? So Amphitropus is near to Anatropus, but in Anatropus the distance became even very less. Here some distance is there, right? But in case of Anatropus, the distance is going to reduce even more and embryo sac is not going to become horseshoe. So in case of Amphitropus, both the shape of the ovule and embryo sac becomes horseshoe. So it is known as Amphitropus ovule. Correct? Now, the most common you have done, most primitive you have done. Now, the last type of ovule is Sarcinotropus ovule. This is the most advanced ovule seen in advanced families of angiosperm like Cactaceae, in Opuntia, Plumbaginaceae. Correct? So, what is Sarcinotropus? It is the most advanced ovule seen in advanced families of angiosperm like Opuntia. Opuntia is a member of Cactaceae, Plumbaginaceae. What happens over here, beta? The funicle, the ovule is orthotropous, straight, but the funicle turns 360 degree around this orthotropous ovule. So what are you going to write? Funicle turns 360 degree along the straight ovule. I'll show you with the help of the diagram. It will become very more easy to understand. So first you're going to draw a straight ovule. Remember the LIC logo, right? So it is going to have micropyle at the top, integuments. This is micropyle, this is chalaza, and this is funicle. So how is it different? Beta, this funicle is now going to turn 360 degree around this ovule like this. Understood? Shall I draw it again? It is looking a little bit horrible. So let me draw it again for you. So first we are going to draw a orthotropous ovule, right? See how. Cute sa little small sa diagram we'll draw. We won't draw it huge. I won't scare you with this diagram. Right? So this is your micropyle. This is embryo sac. This is chalaza. This is funicle. But this is normal orthotropous. How will you show it cercenotropous? What happens in cercenotropous, beta? This funicle is going to turn 360 degree around your ovule. So this is the most advanced ovule, which is known as cercenotropous. I hope all the 6 types of ovule and their examples are clear. Now moving ahead with the main thing which we have to do today is megasporogenesis. Megasporogenesis is the process by which a diploid megaspore cell, megaspore mother cell, undergoes reduction division to produce haploid megaspores. These haploid megaspores then beta develop into female gametophyte, that is embryo sac. This embryo sac is going to carry the female gamete, which will fertilize with the male gamete to start the new generation. Correct? So what is megasporogenesis? It is the event of formation of haploid megaspore from diploid spore mother cell by meiosis within the ovule. So ovule is the main site of megasporogenesis. So if question comes, what is the location of megasporogenesis? You will say ovule. So suppose this is the ovule, the most common type, which is your anatropous. This is the micropyle. This is the chalaza, and it is all filled with nucellus. Now, what happens beta? Within the nucellus, one of the cell of the nucellus towards micropylar end, one of the nucellus grows in size and start behaving as megaspore mother cell. So what I am saying, that one of the cell of the nucellus towards micropylar side, let me write, one of the nucellar cell towards the chalaza or micropyle, towards micropylar end, grows in size and behaves as diploid megaspore mother cell. Right? Now, take out this megaspore mother cell outside. What will happen beta? This diploid megaspore mother cell will now undergo reduction division to produce 4 haploid megaspores. How many? 4 haploid megaspores, out of which generally 3 gets degenerated and 1 remains functional. So let me degenerate 3. This is micropylar end, this is chalazal end. Correct? 3 will degenerate. Maybe from 3 micropylar may get degenerate, or maybe from chalazal end 3 may get degenerate. However, 1 is only going to remain functional, and that one functional megaspore will undergo mitosis to form complete embryo sac, which is your female gametophyte. That is embryo sac. Correct? Now, you can see beta, 4 megaspores are produced after meiosis. However, only 1 is functional, 3 gets degenerated. That means this type of embryo sac formation is monosporic. What is this beta? This is monosporic development. Why? Because there is only one functional megaspore which is undergoing mitosis to produce the complete female gametophyte, female gamete carrying body. So one functional megaspore, monosporic. This is the usual condition. In some cases, two functional megaspore can be there. So bisporic development. In some cases, all the four megaspore may remain functional to form the embryo sac. So that is tetrasporic. So how many types of embryo sac development can be there depending upon the functional megaspore? 3. Monosporic, bisporic and tetrasporic. Getting my point or not? Yes or no? This concept of monosporic, bisporic, tetrasporic was explained by our Indian scientist P. Maheshwari. Understanding? Once again I am repeating, please pay attention. You are having ovule. You know the structure of ovule. One of the cell of the nucellus towards micropylar end. It grows in size, behaves as megaspore mother cell. This megaspore mother cell, my dear students, will then undergo reduction division to produce how many? 4 haploid megaspore. Now, maybe one megaspore may remain functional to form the female gametophyte later on, maybe 2, maybe 4. Depending on this, there are 3 types of embryo sac development explained by Maheshwari. He's an Indian-born scientist, also known as father of embryology. This scientist did a lot in the field of angiospermic reproduction, in the field of embryology. Correct? So, types of megaspore development can be how many? 3. Depending upon the involvement, depending upon the number of megaspores involved in this process. There can be 3 types: monosporic, bisporic and tetrasporic. Correct? Monosporic means one functional megaspore. Bisporic means 2 functional megaspore, and tetra means when all the 4 megaspore remains functional to form the female haploid gametophyte. In our syllabus is the only monosporic one. Bisporic and tetrasporic we are not going to do. We will only focus on monosporic development. In monosporic development, again there can be 2 types. There can be 2 types. One is Polygonum type of embryo sac and second is Oenothera type of embryo sac. So first we will see what is Polygonum and then we will see what is Oenothera. Polygonum is the monosporic is most common. In monosporic, Polygonum type of embryo sac is most common. Now what happens in this beta? See, you were having a diploid megaspore mother cell, correct? Towards micropylar side. It undergoes reduction division to produce 4 haploid megaspore. Now 3 towards micropylar end. Please remember this thing, which one megaspore is functional. 3 towards micropylar end gets degenerated. In Polygonum, again I am repeating, please pay attention. The 3 megaspore towards micropylar end will get degenerated and the one towards chalaza will remain functional. So the chalazal functional megaspore is used. So this is chalazal functional megaspore will undergo reduction division. Now this is going to undergo mitosis. 7 rounds of mitotic division and 3 rounds of mitotic generation. So this functional chalazal megaspore will undergo 7 rounds of, I'll show it with the help of diagram also, but first let me write mitotic division and 3 rounds of mitotic generation to finally produce a 7-celled 8-nucleate embryo sac, right? 7-celled 8-nucleate embryo sac, which is most common type, Polygonum type, right? Now let me show you that how this is happening. Chalo. So you are having one functional chalazal megaspore. What will happen? First, it is going to undergo division as well as first generation. So first division plus first generation. क्या बनेगा? What it is going to produce? 2 nuclei. Acha, okay. Beta, initially it is free nuclear division. Free nuclear means repeated karyokinesis will take place and cytokinesis will happen at the last. So this important point, let me write it over here. This is actually showing free nuclear mitotic division. It's free nuclear mitotic division. So only karyokinesis will take place. Now first division took place, 2 nuclei. So these two are 2 nuclei per cell. No cytokinesis. Now it will enter into second generation, second mitotic generation, and this nuclei beta it will undergo mitosis, right? And this is also undergoing mitosis. So overall 2 more mitotic division. First division already took place. Now 2 more mitotic divisions. So that means in total 3 mitotic divisions. What will the condition? 4 nuclei per cell. Multinucleate condition. Only karyokinesis. Now all these 4 cells will undergo all these 4 nuclei, not cells, all these 4 nuclei will undergo karyokinesis to produce 4 more nuclei. Overall, the cell is undergoing mitosis to third generation. And how many mitotic division? 3 already took place. 4 more to take place. So total 7. So 7 mitotic division. What will be the condition finally? 8 nuclei per cell. Right? Now, all these 8 nuclei will arrange. 3 will come at the center. 3 will come towards the lower side. 3 will go towards the upper side and 2 nuclei will remain in the center. Now cytokinesis will take place. At last, cytokinesis. Right now it was only free nuclear division, only karyokinesis. Now cytokinesis will take place, due to which 3 cells will go towards the chalazal side. This is chalazal side, right? And this is micropylar side. Actually, ovule is anatropous. The most common ovule is anatropous. So we will consider that micropyle is at the base and chalaza is at the top. You know the condition is reverse. But because we are studying the anatropous ovule, hence we are considering that micropyle is at the base and chalaza is at the top. So 3 nuclei they are going to go towards the chalaza. There will be cytokinesis and all the 3 nuclei will separate as 3 independent cells. They are known as antipodals. Non-functional. They are just for decorative items. Right? 3 out of remaining will come at the base. That is towards the micropyle, not the base. But actually with respect to diagram, base, correct? And they are going to undergo cytokinesis. The nuclei will separate as cells. Now, together known as egg apparatus. So, how many now cells are there? 6. Egg apparatus consists of one egg, the upper one, and 2 lower are called as synergids. Yes or no? Okay. Now, how many remaining are there, beta? 2 are remaining. Those 2 remaining nuclei will come in the center, right? The independent, they are called as polar nuclei. Later on, beta, they fuse to form secondary cell. Correct? Yes or no? So, how many cells? 7 cells. 3 antipodal, 1 central, 3 lower. But how many nuclei? 8 nuclei. 3 antipodal, 2 secondary, 3 egg apparatus. So, this is 7-celled, 8 nucleate condition of embryo sac, most common Polygonum type. Now, is it clear to each and every one of you? Yes, very good. Beta, now we'll study all these individual, some of the properties. Let's start with egg apparatus first. Okay, now Polygonum type in detail. First, we are going to start with egg apparatus. Egg apparatus consists of first synergids. Synergids, beta, they are also known as helper cells, right? Also known as cooperative cells. How many number? 2. Now, synergids, if I specifically talk about, let me draw it over here. This is the egg apparatus, 3-celled, 3-nucleate condition egg apparatus. This is the micropylar end, this is the chalazal end. Now, synergids, these two are synergids. Synergids, beta, on their micropylar end, they have some finger-like projections called filiform apparatus. So, they possess, first point, micropylar filiform apparatus. What is the role? When pollen tube will come, this filiform apparatus will help in the entry of pollen tube. So yes, it is going to help in the entry of pollen tube when the right time will come. Now, towards the micropylar end, beta, they have nucleus. And towards the chalazal end, they have larger vacuole. So this is their vacuole and this is their nucleus. So what will you write? That filiform apparatus, beta, they have chalazal vacuole and micropylar nucleus. Right? It is also said that egg and synergids, they are connected with the help of plasmodesmata. So there are plasmodesmatal connections between the egg cell and synergids. Why? Because the synergids are actually providing nutrition to the egg, and that is why this egg is developing. So the source of nutrition for the egg is the synergids. That is why they are called as cooperative cells. Correct? Now, one more thing, it is said that, beta, towards the chalazal end, these synergids, they lack cell wall so that they easily connect with the egg. So you can write this point as well. Towards chalazal end, synergids lacks, they degenerate their cell wall so that they can easily establish connection with the egg. Now coming to the next female gamete, which is non-motile egg. Egg is supported by two synergids which are connected with the help of plasmodesmata. Now the polarity of cytoplasm, the polarity of vacuole and nucleus in case of egg is just the reverse. That means these egg will have vacuole towards the micropyle and nucleus towards the chalaza. Correct? Yes or no? Okay, so egg is having just the reverse polarity, that is they have chalazal micropyle, they have chalazal nucleus and vacuole is towards the chalaza. So this is vacuole. So we can write the polarity is just the reverse. That means they have chalazal nucleus and micropylar vacuole. This is the actually cell which fertilizes with the male gamete to form zygote. Understood? The egg apparatus, which is overall 3-celled, 3-nucleate. Remember, sometimes question is asked on this only. And students just remember 7-celled, 8-nucleate and they just tick it without looking at the question that it is asking for egg apparatus, not for the whole embryo sac. Right? Chalo. Now, moving ahead, egg apparatus is done. Wait, let me add a page. Okay, after this, next are your central cell. Now, central cell is what, beta? Also called as definitive cell. These are the only diploid cell of the embryo sac present at the base of the egg apparatus. Now, in the diagram, you will see that they are present above the egg apparatus. However, you know that the diagram is placed in a reverse manner. Actually, in real sense, if you keep the embryo sac upright, you will see egg apparatus on the top, then central cell and then antipodals. However, it is kept in inverted manner. So what happens? Antipodals come at top and egg apparatus comes below. But what is actual real condition? Central cell are the diploid cell formed by the fusion of two polar nuclei. Right? Plus, they are present at the base of egg apparatus and top of antipodals in real sense. So, don't go with the diagram for the position. And top of antipodals. I hope you understood the point what I am trying to say. Okay, now later on this central cell, the secondary cell will fuse with the second male gamete to form the triploid endosperm. But that point I am not going to write it over here because when I'll teach you double fertilization concept, there I will tell you. For here, this much is important. Last are antipodals. Antipodals are usually present on the side of chalaza. So their chalazal end, they are present on the chalazal end, number 1 point. Second, they are 3 in number with no function. No function allotted to them. They are just the decorative pieces. Soon after fertilization, they degenerate. No as such special function assigned to them. I hope Polygonum type of embryo sac is clear to each and every one of you. If yes, then let's start with the second type of monosporic development, that is Oenothera type. Oenothera, very common, not that common, common in some specific families. Now what happens over here? Beta, this is one functional megaspore, right? What happens? This functional, first this is my diploid megaspore mother cell. Okay, bacha, it will undergo reduction division. What will happen? 4 haploid megaspores will be produced. How many? 4. Micropylar, chalazal. Now, for Oenothera type of development beta, 3 chalazal megaspores they get degenerated and 1 functional micropylar megaspore persist. Just the reverse of Polygonum. In Polygonum, you saw that functional megaspore is chalazal. In case of Oenothera, the functional megaspore is micropylar. 3 megaspore which gets degenerated are from the chalaza. So this is the micropylar functional megaspore, right? Which is now going to undergo reduction division. Which is now going to undergo mitosis. How many mitosis? It is going to undergo 3 rounds of mitotic division and 2 rounds of mitotic generation to produce 4-celled 4-nucleate embryo sac with 3 mitotic divisions and 2 mitotic generation. Now let's check out how is this happening. Now this is beta my functional megaspore, haploid megaspore, right? Free nuclear division. Remember here also free nuclear division. Only first karyokinesis, cytokinesis will take place but at last. So this is the nuclei. It is going to undergo first round of division as well as first round of mitotic generation. From one nucleus, it is going to become binucleate, multinucleate, 2 nuclei per cell. As no cytokinesis is first taking place. Now, these 2 nuclei will again undergo karyokinesis, second division, third division, but generation overall will be 1. So, second generation, right? And 3 mitotic division, first already took place, 2 over here will happen. So, total 3 mitotic division free nuclear. How many cells will come now? From 2, the number of nuclei per cell will become 4. Now cytokinesis will take place. After so many divisions, now cytokinesis at last will take place. Out of 4 cell beta, 3 comes below towards micropyle and forms the egg apparatus in Oenothera type. Getting my point? Which is again 3-celled and 3 nucleate. Now how many cells are left? 1. How many nuclei is left? 1. Now this one is going to come in the center showing 1 polar nuclei. No formation of secondary cell, no antipodals in Oenothera type. This is 4-celled, 4-nucleate Oenothera type of embryo sac which consists of 1 polar cell, 1 polar nuclei and 3 egg apparatus. No formation of secondary cell, no formation of antipodals. So remember in Oenothera type, note point, there are no antipodals. Now there is no function of antipodals also. Hence no trouble, no problem, no antipodals. It's okay. So no antipodals and second beta, you know that since secondary cell is not formed. Now when male gamete will come and fertilize with this 1 cell, diploid endosperm will be produced instead of triploid. So in Oenothera type of embryo sac, the ploidy of endosperm will be 2n. Reason that central cell is single nucleate. There is no central double nucleate cell as you see normally in Polygonum type, right? So one more male gamete will come, fuse this with single nucleus, double nucleate structure will be formed, that will be called as endosperm, right? So this much is there for monosporic development. I hope you all have understood this so far, that what is types of ovule, what is the concept of megasporogenesis, and types of monosporic development with the details. If yes, then we are going to keep our lecture for today till here. You have some homework as the tradition says, homework number 1 and homework number 2. Please try to solve them, and as usual write the answer in the comment section beta. Very important, take the classes very seriously. Please, it's a humble request. Share the lecture with your friends, share it with everyone, let everybody get benefit. When the benefit is being spread, let everybody take it. So thank you for watching. See you in the next class. Bye bye everyone, take care.